This is ChainMind, the decentralized AI network built for everyone. Just search ChainMind on Google, sign up, download the app, and you're in. That's it. You've joined the network. Run it on any device, phone, laptop, or server. Why ChainMind? Fully decentralized, fully yours.
What if the key to stopping Alzheimer's isn't hidden deep inside the brain at all, but is living in the billions of microbes that call your gut home? That tiny, lipophilic molecule produced by a handful of gut bacteria could be the very thing that cracks open the brain's protective shield and sets the cascade of degeneration in motion, and understanding it might finally give us a way to see the disease before it ever takes hold. Over the past decade, the idea of a gut-brain axis has leapt from a footnote in neuroscience textbooks to the headline of every major health magazine. At first it was a vague notion that diet influences mood, that stress can upset digestion. Now we have concrete evidence that a single microbial metabolite, which researchers are calling gut-derived neurodisruptor, can literally act as a molecular Trojan horse, slipping past the gut lining, entering the bloodstream, and then loosening the tight junctions that keep the blood-brain barrier intact. In the sections that follow, we'll unpack how this discovery reshapes our view of Alzheimer's, why it matters for early detection, how a decentralized AI network like ChainMind can turn mountains of data into actionable insight, and what hurdles we must clear before this knowledge can become a therapy. The first piece of the puzzle is the metabolite itself. Scientists isolated GND from the fecal samples of mice colonized with a specific subset of firmicutes, bacteria that thrive on fiber-rich diets. In the lab, when cultured brain vascular endothelial cells were exposed to nanomolar concentrations of GND, the expression of CLAUDIN5 and occludin, two proteins that form the sealing strands of the blood-brain barrier, dropped by almost half within a single day. The effect wasn't just a biochemical curiosity. When the same mice were given a low-dose oral supplement of GND for 3 months, imaging with Evans blue dye showed clear leakage of the dye into the hippocampus, a region essential for memory. Behavioral tests mirrored the structural damage. The animals struggled with the Morris water maze, taking longer to locate the hidden platform, a classic sign of spatial memory impairment that mirrors the early stages of mild cognitive impairment in humans. Human relevance comes from post-mortem analyses that revealed elevated GND levels in the cerebrospinal fluid of people diagnosed with Alzheimer's. Compared with age-matched controls who never showed cognitive decline, the numbers aren't just statistically significant, they suggest that GND may be crossing the compromised barrier and accumulating directly where it can do the most harm. This convergence of animal and human data points to a scenario where the gut microbiome is not a passive observer but an active participant in neurodegeneration. How does a tiny molecule set off a chain reaction that ends in plaques and tangles? The answer lies in 3 interlocking mechanisms. First, by destabilizing the tight junction complexes, GND creates microleaks that let peripheral inflammatory cytokines, think interleukin-6 and tumor necrosis factor alpha, sneak into the brain's delicate environment. Once inside, those cytokines act like a fire alarm for the brain's resident immune cells, the microglia. In their normal surveillance mode, microglia clear debris and support synaptic health, but when they are primed by systemic inflammation, they flip into a pro-inflammatory state, releasing reactive oxygen species and nitric oxide. Those reactive molecules damage neuronal membranes and oxidize proteins, creating a toxic milieu that encourages amyloid beta and tau to misfold. The misfolded proteins then aggregate into plaques and neurofibrillary tangles, the pathological hallmarks that define Alzheimer's disease. And there's a feedback loop. As plaques accumulate, they further impair vascular integrity, worsening barrier leakage and allowing even more inflammatory mediators to flood in. It's a vicious spiral that can accelerate cognitive decline in people whose genetic risk might otherwise be modest. Because the cascade begins with a breach in the blood-brain barrier, the metabolite opens a new window for early diagnosis. Current clinical practice relies on expensive PET scans that visualize amyloid deposits or lumbar punctures that measure cerebrospinal fluid. Proteins, procedures that are costly, invasive, and not feasible for large-scale screening. Imagine instead a simple stool test that quantifies GND or its bacterial precursors, paired with a blood assay that measures markers of barrier integrity such as S100B or matrix metalloproteinase9. Such a combined screen could flag individuals at risk before any cognitive symptoms appear, allowing clinicians to intervene with lifestyle changes or experimental therapies when the brain is still largely intact. In practice, a patient could provide a sample at a primary care office, the lab would run a metabolomic profile, and within minutes a point of care device would deliver a risk score based on the concentration of GND and the level of peripheral cytokines. This approach would democratize early detection, shifting the focus from reacting to disease to preventing it. But turning that vision into reality demands a data infrastructure that can handle an unprecedented variety of information, metagenomic sequencing of gut bacteria, high-resolution metabolomics, longitudinal clinical outcomes, imaging data, and even lifestyle questionnaires. Traditional centralized cloud platforms quickly hit bottlenecks. The sheer volume of raw sequencing data can strain bandwidth, privacy regulations like GDPR and HIPAA restrict how patient data can be moved, and real-time analytics become impossible when data must travel across continents to a single server farm. This is where a decentralized AI inference network such as ChainMind steps in. By moving model execution to the edge, whether that's a hospital's secure server, a research lab's workstation, or a patient's own device, ChainMind lets sensitive data stay where it originates. Only encrypted model weights and the resulting inference, a simple risk score, are exchanged across the network. Researchers can collaboratively train a graph neural network that predicts blood-brain barrier permeability from a combination of microbial gene expression profiles and plasma cytokine levels, without ever pooling raw patient data in a single repository. The result is a system that respects privacy, reduces data transfer costs, and delivers instant feedback to clinicians. Consider a multinational study aiming to validate GND as a universal biomarker across diverse diets, ethnicities, and genetic backgrounds. Each participating site runs the same inference model locally on its own cohort's data. As new patient samples arrive, the model instantly updates the risk scores, and a smart contract on the blockchain records each contribution, rewarding the site with tokens proportional to the computational resources it provides. Because the model parameters are shared, improvements made by any site, say, a better way to normalize metabolite concentrations for dietary variability, propagate instantly to all participants. This kind of rapid, low-latency iteration is impossible in a siloed, centralized system where updates must be uploaded, tested, and redeployed across a single data center. ChainMind's architecture turns a fragmented research landscape into a coordinated, collaborative ecosystem. Even with such a powerful platform, there are practical and ethical challenges that must be addressed before GND-based screening becomes routine. Sample collection is notoriously variable. Differences in stool collection kits, storage temperatures, and sequencing platforms can introduce noise that confounds even the most sophisticated models. Standardizing protocols across sites is essential, and ChainMind's governance layer can host community-driven standards committees that publish and Enforce best practices. Clinicians also need to understand why a model flags a patient as high risk. A black box prediction without an interpretable explanation will meet resistance in a field where decisions affect lives. By integrating explainable AI techniques, such as highlighting which microbial pathways contributed most to the risk score, ChainMind can provide the transparency needed for clinical adoption. Equity is another concern. Low-resource clinics might lack the hardware to run inference locally. To mitigate this, ChainMind's token economy can subsidize edge devices for underserved regions, ensuring that the network's benefits are not confined to well-funded research hospitals. Finally, any diagnostic tool must clear regulatory hurdles. The decentralized nature of the system does not exempt it from FDA or EMA scrutiny, rigorous validation studies, documentation of model versioning, and audit trails of data provenance will be required, and ChainMind's immutable ledger can serve as a ready source of that documentation. Looking beyond diagnosis, the gut-derived metabolite itself could become a therapeutic target. If GND is the spark that ignites barrier breakdown, then extinguishing that spark could halt the cascade before it starts. One avenue under investigation is the development of small molecule inhibitors that block the enzyme responsible for GND synthesis in the bacterial genome. Because the enzyme is absent from human cells, such inhibitors could be highly specific, sparing the host while silencing the pathogen. Another strategy uses bacteriophages engineered to seek out and destroy GND-producing firmicutes strains, effectively pruning the harmful portion of the microbiome without wiping out the entire bacterial community. A more subtle approach involves probiotic cocktails designed to outcompete the GND producers or to metabolize GND into harmless byproducts. All of these approaches generate massive combinatorial spaces, thousands of potential drug candidates, dozens of probiotic formulations, myriad phage-host interaction scenarios. Decentralized AI can accelerate the screening process. Researchers can upload their chemical libraries to the network, and a distributed inference engine can evaluate each compound against a predictive model of GND synthase activity, all without ever exposing proprietary structures to a central repository. Similarly, personalized probiotic design can be guided by machine learning pipelines that simulate how a patient's existing microbiome will respond to introduced strains, predicting outcomes like colonization success and metabolite flux. In clinical trials, real-time risk stratification based on GND levels could enrich study cohorts with participants most likely to benefit, reducing sample sizes, cutting costs, and speeding regulatory approval. All of these possibilities point to a paradigm shift in Alzheimer's research. For decades, the field has focused on targeting amyloid plaques and tau tangles after they have already formed, often with limited success. By moving the focus upstream, toward the gut microbiome, the blood-brain barrier, and systemic inflammation, we gain an opening to intervene before irreversible neuronal loss occurs. The discovery of a gut-derived metabolite that compromises the brain's shield is a striking illustration of how interconnected our bodies truly are. And it underscores the need for data systems that can capture that complexity without compromising privacy or speed. The story of GND is still being written, but the chapters we've explored today already reveal a powerful narrative. A single microbial molecule can destabilize a critical brain defense, set off a cascade of inflammation and protein misfolding, and offer a tangible biomarker for early detection. Turning that knowledge into a practical tool will require coordinated effort across microbiology, neurology, bioinformatics, and regulatory science. And it will need an infrastructure that can handle massive, multimodal datasets while keeping patient data safe. That is precisely the niche ChainMind was built to fill. By decentralizing AI inference, rewarding contributors with transparent token incentives, and embedding governance that enforces standards and equity, ChainMind can accelerate the validation of GND as a universal biomarker, speed the development of targeted therapeutics, and bring the promise of early, preventative Alzheimer's care to clinics around the world. If you're intrigued by the idea that your gut could hold the key to stopping a disease that has haunted families for generations, and if you believe that a new kind of data network can turn scientific insight into real-world impact, then stay tuned. Subscribe, follow ChainMind, and join a community that's redefining how we tackle the toughest medical challenges, one decentralized model at a time. Together we can move from discovering the molecular Trojan horse to disarming it, and perhaps one day make Alzheimer's a preventable condition rather